So then we know that that's demonstrated great gains in areas in which speed is key. But I want to hear from you both. What are the most important gains required to, extend the use cases for the technology to bring it to billions more? Meaning, what are the therapeutics or applications that are most likely to be accessible with the current cohort of the next generation technology? Nathaniel, can we start with you for that one?
Yeah, definitely. In terms of what the technology needs to do, from my perspective, we need to boost the bioactivity, and that's what we're focused on a lot. So if you can drop the doses by 1,000-fold or 1,000,000-fold and still be getting the same biological effect, that opens up a lot more applications in areas like protein drug replacement. But if we think about what are going to be the most accessible areas, I think we look at where the stretch cases of the technology are currently being identified. So for instance, we've seen the technology work very well for RSV. It will probably be seasonal. We look at some of the trials out there in influenza, where 2 out of 4 of the seasonal strains look good, but the other 2 were more or less misses. And you can start to see that, you can't go higher in doses. It's a little bit difficult to hit those metrics. And we see things like personalized cancer vaccines that I had mentioned before, which, as the name implies, are very personalized, can be very expensive, and require a lot of challenges in manufacturing. If you look at the nearest adjacencies for all of those areas, infectious disease, it's more complex infectious disease. So whether you're looking at highly multivalent flu vaccines, whether you're looking at complex infectious diseases where you may need a lot of pathogen proteins, things like Epstein-Barr virus and things like that, which have recently been linked to multiple sclerosis and Parkinson's, and are pretty complex in terms of how you would want to go about targeting them. That's one example of a near-term area. Another one is in the area of directed cancer therapies, whether it's cancer vaccines or things of that nature. The success of the recent Merck and Moderna data in melanoma has been really impressive, but building off of those into more off-the-shelf approaches that tackle cancers that have an even higher prevalence and hit more patients, I think is something that the field is looking to do. And then I've mentioned the term protein drug replacement before, which is just a fancy term for having the protein produced in the cells in your body. And I think that that's probably the next closest area, and probably areas like inflammatory diseases, where they're validated pathways. We know what molecules we need to go after, but we know that the profile that linear mRNA hits isn't sufficient. We're going to need more protein. We're going to need it for longer to be able to soak up some of those inflammatory molecules that really underpin a lot of autoimmune diseases. So that's probably one of the next nearest after the other 2 areas. So, that's how we see it. But, the great part about emergent technologies and emergent spaces is there's plenty of room for different points of view and for all of us to be right. So it's not a zero-sum game.
For sure, for sure. Sam, as Nathaniel mentioned, as we're moving to advance the technology into these more challenging areas, from your perspective, what would be required from an engineering perspective to advance the technology to, in fact, do that?
Yeah, I think it, as Nathaniel was pointing out, it really depends. So the engineering that you need to achieve, again, depends on your mechanism of action and what you try to achieve and what the TPP for a specific product is going to be. I think, as Nathaniel said, that infectious diseases, some aspects of immune system modulation, where small perturbation can lead to longer lasting effects by training the immune system to either go after an antigen or develop tolerance against an antigen are different axes of kind of the same way in which you can use mRNA either for infectious disease, cancer, or for autoimmune diseases. I think the next stage will be two aspects of protein replacement. So one is really strictly protein replacement, where you want to supplement the body to overcome some sort of metabolic disease or enzyme deficiency, or situations where you want to produce a therapeutic protein using the body cells. So, for example, producing a monoclonal antibody or a bispecific or a trispecific. There are certain situations where it could be very advantageous to have your own body produce it rather than give it as a bolus injection. I think on the longer horizon, things like RNA-based gene editing or in vivo CAR T cells or other types of in vivo cell therapies, and eventually regenerative medicine applications are actually very exciting. Some of these things, as we said before, are still autonomous and require a very high degree of mRNA engineering, and to be honest, still some tech development to allow us to get there. And I think a lot of our thinking on Nutcracker is to really understand that interaction between our formulations and biological systems in vivo. How can we, for example, in the situation of protein replacement therapy, where we think we're going to be dosing chronically, can we dose at intervals that do not impact the cell health, that do not lead to lipid accumulation that could have long-lasting effects? So how do we engineer faster clearances, higher tolerability, low immunogenicity into our formulations? I think will be very important aspects of this, and very enabling to open up the mRNA therapeutic space to this new set of indications that are out there.
Good. Okay. Thank you. We have time for about one audience question. So thank you to our audience for submitting them. And this audience member is asking, how do you see the manufacturing being managed for personalized therapeutics? Sam, since you're at the mic, if we could start with you for that response.
Yeah, I think an important aspect of this is scalability and automation, right? When you're making this N of 1 type drugs, if you imagine having a team of 5 or 6 or 10 people that are dedicated to the production of a drug for a single patient, then we're going to end up in kind of the same setting as this personalized autologous CAR T therapies that end up costing $1 million or $500,000 per treatment, right? So how do we move away from that paradigm to a paradigm where these things can actually be reasonable in terms of cost, but also scalable? And our approach to this is really through this microfluidic automation systems, where a single person can be working on 10 of these NMU machines that and each one producing drug for an individual patient, where a lot of the quality control is done in real time, also through automation. So also the release and QC aspect of this, which is very costly, can also be somewhat controlled. And there's always going to have to be some potency, identity, and sterility aspects to the release of a product that you have to do, no matter if it's for off the shelf or personalized. You always have to do those things. But a lot of the other costs can be mitigated through automation and control. So that's the way that we see it. That for us to really be able to execute on personalized settings at a larger scale, will require this very controlled, very reproducible systems.
For sure. And Nathaniel, I want to give you the opportunity to respond to that question as well. What are your thoughts?
Yeah, absolutely. And so, when you're doing N of 1 manufacturing or for an individual patient, as Sam had mentioned, you're already obviously working with a lot less material than if you're doing it for a lot of patients. And if we start looking at some of these next gen techs where, in our case, we're having 1,000th or 1,000,000th of the dose, then that becomes really small amounts of material. And it's funny, as we talk to our partners on the manufacturing side, people are always being pushed on the manufacturing side. How much can we scale? How big can we make it? And, all of those testings that you need to do on the material that Sam was mentioning was really built assuming that you have this giant scale and it's easy to just pull off a little bit of the material. Now we're pushing them in the exact opposite direction in a way that they haven't thought about maybe in years or perhaps ever, where it's like, okay, how small can we make this? Like, how small of a bioreactor can we make, and can we release material? And the reality is, because a lot of these assays and these tests still need to be run with a pretty substantive amount of material, and because you need to serially test things for stability or these other types of tests, it isn't a small material you need anymore, even though it's just for a single patient. And so, in those cases, the cost may not even drop a lot, even though we've dropped the effective dose, because you still need that amount of material. And it's the testing that ends up driving the lot size, as opposed to the actual clinical dosing that drives the lot size. And that's just a very different way to think. And so that's why you end up getting these personalized therapies, even for RNA cancer vaccines, that end up costing in the hundreds of thousands.